Good evening, everyone. We're so glad you're here tonight. Welcome to Science Cafe. My name is Jenny King, and I work in public affairs here at UT Southwestern Medical Center in Dallas. Our team is proud to produce and host the UT Southwestern Science Cafe series. Along with my co-host, Charlie Thompson, our public affairs team member Jovelyn Castellanos, our summer intern, Erin Renfro, as well as tonight's speaker and our many colleagues, we thank you for joining us. Science Cafes are online conversations where speakers take you on deep dives into the science behind healthcare. As an academic medical center, UT Southwestern brings together research, health education, and patient care into one institution. Tonight's program explores the connection between gut microbiota and the brain, the effect that certain microbes can have on your mood, and the global research on this subject. Before we start the program, I have a few housekeeping items to share. We are recording Science Cafe as well as live streaming it on our Facebook page. Please mute your microphones so we can have good audio for everyone. As we are all connecting digitally through Zoom, help us build community by considering leaving your camera on during the Q&A session so we can see each other. And finally, while we cannot answer personal medical questions, we welcome your general questions about tonight's topic. Throughout the next hour, please list your questions in the chat. Charlie will address them after our presentation. And now I'm so pleased to introduce our speaker. Dr. Jane Foster is Professor of Psychiatry at the Center of Depression Research and Clinical Care here at UT Southwestern. As a neuroscientist and leading expert on the microbiome, Dr. Foster joined UT Southwestern last year to lead the effort to connect the dots between a person's 30 trillion gut microbes and their propensity for mental illness. By combining basic science research with clinical collaboration in psychiatry, psychology, and gastroenterology, Dr. Foster's research takes a bench to bedside and back again approach to studying microbiota, brain, and immune brain systems. Dr. Foster received her Ph.D. in 1996 from the University of Toronto. She trained as a postdoctoral fellow at Henry Ford Health System in Michigan and as a research fellow for the National Institutes of Health in Maryland from 1999 to 2003. After that, she was affiliated with McMaster University until joining UT Southwestern in 2022. Please click on Dr. Foster's bio link and her research lab link, which are being shared in our chat, to learn more information about her research, publications, and honors. And now, Dr. Foster, welcome to Science Cafe. The virtual podium is yours.
Thank you. Let me see if I can. Okay.
Excellent.
I see there's more people in the waiting room. I assume somebody's taking care of that.
They come in all hour long and we will let them in.
Okay, I'm just trying to see if I can make you guys look smaller.
And folks, we may go a few minutes over depending on our questions because we started a minute late. And so I just wanted you to know that.
Okay. So thanks for the invitation and thanks for everybody joining. I just thought I'd introduce a little bit about me. Much my bio was shared, and as we say, Foster Lab 1.0, which was in Hamilton, Ontario for 19 years, moved to Dallas, Texas, about this time last year. So I'm going to first introduce the microbiome. Some people might know a little bit about it. Some people might know a lot about it, but we're just going to all get on the same page. And then I'm going to tell you about some translational research that links microbes to anxiety. So there's been a lot of work in this area over the past 15 to 20 years, and it's really increased in the clinical space in the last 5 years. And then I'm going to talk about potential approaches we might use to leverage the microbiome to have an impact in psychiatry. And we'll talk a little bit more about that as we go. So in the past decade, there's been a paradigm shift in brain health from physicians and researchers thinking from a brain-centric approach and moving more towards a holistic approach. And that's represented here in this cover from a review article we wrote for the Trends in Neuroscience about a decade ago, suggesting that we actually have to think of the brain as a component of the holistic system, and it's in continual interaction not only with the body, but within within the environment. And from a medical school perspective or a treatment perspective, this has not been the general approach that people have used for psychiatry, and it's probably an important shift that needs to happen. So just to let you know about microbes, they cover all the surface of your body, and they include things like bacteria, viruses, fungi, protozoa, and parasites. The composition and the diversity of your microbes varies between the different body sites, which is shown here in this little graph on the right-hand side from the Human Genome Project, showing that in 300 individuals, there's differences within yourself between your gut bacteria and your bacteria in your mouth, and even between individuals. And from a numbers game, there's actually 100 trillion gut bacteria in an adult human, 1 trillion bacteria on the skin, and 10 million bacteria in your mouth. So when you do the math, you're actually 1.6 times more bacteria than you are human. And what's remarkable is the biochemical potential of your microbiome, because as we know from the Human Genome Project, we have about 23,000 genes, and our bacteria have at least 2 million, if not more, genes. And so these numbers in this biochemical potential really point to the reason that these things might have a purpose in our health. And so one of the things that the microbiome does is that the good bacteria fight off the bad bacteria, so they're important to pathogen defense, but they're also very important to nutrient uptake and metabolism. And without them, we would not be able to digest a lot of the insoluble fibers that we put into our through our diet. They're very important to maintaining the integrity of your gut barrier, and there's a lot of crosstalk that happens between your microbes and your barrier systems. They're essential to the development and function of your immune system, and as we're talking about today, they are important to healthy brain development. So when we think about our microbes, it's important to think about where they came from. And so depending on whether you're born vaginally or by C-section, you either gobble up some microbes as you go down the vaginal tract, or you get microbes in the first few hours of life from the womb or from the breast if you're breastfed. So within this early window of life, there's a lot of changes happening with respect to your microbiome. And once it's seeded, it continues to develop over the first few years of life, with ending up what looks like mostly a adult microbiome by the age of 3 years old. And along the bottom here, you see a lot of different factors that might contribute to the microbiome in a developing child. And this composition and function of your microbiome can in fact influence brain development. And this is visualized on the right-hand side of this slide, where if we think about human brain development, and particularly that first year of life, we know that the brain develops in a sequential manner. So that if you think about how babies' developments, their sensory systems are first, then their language, movement skills, higher cognition. And what's interesting, if you look at the schematic in the bottom right-hand corner from a review out of John Cryan's group, is as your brain is, as these activities are happening, your brain is actually increasing in complexity, your synaptic connections are happening, and there's a lot of maturation and complexity added in at the same time that your gut bacteria are in fact maturing and becoming much more complex. So these things happening in the same time in a developmental window provide this opportunity for a crosstalk between these systems to influence each other. And so I'm going to go a little bit into why we think this might be important. And one of the things about the microbiome is that it's your own. And we know that no two fingerprints are the same. And so we've known about the fingerprint since the late 1800s. And what's interesting is just only this spring, the genetics basis of fingerprints was published. So there's been a lot of discussion. And in fact, your fingerprint develops in utero from epithelial cells, and the details of which are not important. But the difference between your fingerprint, which has been a very important individual marker, and your microbiome, while it's your own, it's actually influenced by your genetics, but it's also influenced by a host of other things, such as diet, exercise, your age, your sex, any antibiotic or non-antibiotic drugs you taste, as well as stress. And so, in fact, your microbiome equals your genes plus your environment plus your life history. And we think that if we take a look at the microbiome at any cross-sectional point in your life, it might actually give us a biological signature of an individual. And in that way, it sort of is a snapshot of not only where you are today, but where you've been from a health perspective up until now in your life. And so the challenge in psychiatry is that we use a whole host of behavioral and clinical tools in order to diagnose people, but these tools don't rely on biology. And in response to first-line antidepressants, such as SSRIs, only a subset of individuals respond. And currently, the tools we have aren't able to let us know when somebody comes into the clinic whether or not they're going to respond to a first-line treatment or whether they need to go down another path. So the hope is that we can use biomarkers or biosignatures in order to take a look at an individual's biology at that onset of disease, or even when they're at risk of disease, in order to guide treatment selection and interventions. So beyond the treatment prediction, there are a whole host of benefits to considering the microbiome in clinical populations. One is to subgroup or stratify clinical populations. One is to screen people for potential microbe host or microbe drug interactions. And then in adolescents or in young adults, potentially, or even later in life, identify individuals at risk for early intervention, potentially provide novel targets for drug development if we start to understand the mechanisms of how the microbes talk through your body to your brain. There's the potential of us developing therapies for anywhere along that path. And then also the idea about expanding and developing diet, prebiotic, probiotic, and other microbiome-related intervention strategies. And I'll go over these closer to the end of the talk. So how does the gut bacteria communicate with the brain? So this is a schematic of the microbiota gut-brain axis. And so those trillions of bacteria are in your gut. And I guess I'm not sure if I mentioned earlier, but most of the work to date has been done looking at gut bacteria, linking it to brain health. And that's mostly because that's where most of your bacteria are, but it's also the place where the interface of the bacteria interacts with things like your diet, your immune system, and other systems. So there's really very important gut-liver communications. This has got a lot to do with metabolism, and there's a lot of health related to that. There is neural connections. One of the nerves that connects your gut bacteria to your brain is your vagus nerve. There's also the stress system, where your brain responds to stress by producing hormones that result in the release of cortisone or cortisol from your adrenal gland. But in addition, your microbes can impact how you respond to stress. And then finally, there's also the immune system. And because your microbiome educates your immune system and regulates over the lifespan, it's possible that the immune cells can in fact communicate with the brain. And so this just shows you a blow up of that area just to orient us all. So this is a little picture of the colon, and you can see the epithelial lining, which are those cells. And within those cell linings, there's a lot of specialized cells. So the orange cells are endochromaffin cells. So these are cells that produce about 95% of the serotonin in your body. And then there's also goblet cells. And then there's within that lamina propria is the whole mucosal immune system. And so this crosstalk at this level is very important to not just to your mucosal immune health, but also your peripheral and systemic immune health. And then we see the enteric nervous system, which are those neurons that are a flat sort of circle embedded in the wall of your gastrointestinal tract that are important not only to motility, but also are connected up to the central nervous system. So I'm going to go over just a couple of papers or findings that take us from where we started, which from my perspective is in 2004, and moves us towards sort of where we first started getting into clinical work. And then I'm going to jump ahead and tell you a bit about some recent work that's actually is the reason I moved to UT Southwestern. And so if we start in the beginning, there's a Japanese group that looked at germ-free mice and published a paper around 2004. And germ-free mice are mice that are the same as all the other research mice or animal study mice that are used, but they lack all microbes. So they live in a sterile system. And what this group showed was that germ-free mice, when exposed to stress, had an exaggerated stress response. And so this drew the attention of myself and other neuroscientists that were looking at this interface of how the body influences the brain. And my group, and about the same time, Rochelle Diaz-Height's group in Stockholm, demonstrated that these mice lacking all bacteria also had reduced anxiety behavior, suggesting that, in fact, the microbes might be important. This was reproduced a few years later by Jer Clark and John Cryan. But also, Chemek Bursic and Steve Collins from McMaster University did a very important experiment. So what they did is they looked at two different types of mice. So mice have genetic differences depending on the inbred strain or the outbred strain you look at. And some of them by nature have higher anxiety and some of them have lower anxiety. And what Chemek did is he took the feces from the low anxiety mouse and gave them to the high anxiety mouse and swapped their phenotype, suggesting this is the early mouse to mouse fecal matter, fecal material transplantation, but suggesting that the microbes themselves contribute to the behavior. And so, as you can see on the left-hand side of this slide, a lot of the early work was done in animal studies. But in fact, it was a decade ago that Emmer Mayer and Kirsten Tillish actually demonstrated that if they fed a fermented milk filled with probiotics to healthy women and did imaging pre and post their consumption of the probiotics, they actually could have an impact on the resting state network that's associated with stress. And so this sort of was the first big story that made people think about that this might actually be translated to people. Okay, so if we fast forward a decade, I'm going to tell you about this paper that was just published earlier this year out of the group here at the Center for Depression Research and Clinical Care at UT Southwestern. And this looked at depressed participants in this study that's called, it's a longitudinal study, naturalistic study called TRAD, which is the Texas Resilience Against Depression study. And there's two arms to the study. One is tracking individuals that either have a current or a previous diagnosis of depression and looking at behavior, physiology, biology, neuroimaging, and physical activity data. And another cohort that looks at youth, which is age 10 to 24, and does the same things, outcomes. And then these individuals come back every 6 months for an extended period. And so the goal of this work is to look at not only the trajectory of depression, but try to start figuring out, if we look at biomarkers, how that relates to a treatment course, but also when can we intervene. And so this study, we took stool samples from 178 individuals, and we isolated the DNA and sequenced the 16S ribosomal RNA gene, which is a microbial gene that is found only in bacteria. So it allows us to determine which bacterial taxa are present. And we also looked at the clinical symptoms in these individuals, including anhedonia, which we measured with the DARS, depression, which we measured with the self-report PHQ-9, and anxiety, which we measured with the GAD-7. And we did it in a fairly novel way. 16S ribosomal RNA sequencing, as again, it tells you the composition of an individual's stool, but there ends up giving you several hundred individual sequences representing different taxa. And so there's been a lot of work looking at the differences between depressed people or healthy people to identify what bacteria might be important. But in this case, we borrowed a tool from the genomics people. We actually looked at the community's bacteria. And what we did, although it's hard to understand if you don't know the math, we identified in this depressed cohort that there were 3 communities of bacteria that were co-occurring. So these are networks of bacteria that we could then generate a value to represent an individual's, in this case, what we call brown, blue, and turquoise module, which are communities of bacteria. And when we looked at these communities of bacteria, we found that this brown module, which is a group of 86 or 87 bacteria, actually were associated with clinical symptoms, including anxiety, depression, and anhedonia. And we can use these tools, analytical tools, to identify which bacteria are most connected in these networks. And when we did that, we identified these hub taxa, which were actually butyrate-producing bacteria. And the way it looked was that individuals less of these key, what we think are protective bacteria, had higher levels of anxiety. And so butyrate is a short chain fatty acid, which is important to your immune health and to your barrier integrity. And it's produced by fermentation of fiber by your gut bacteria. And so this is one of those nutrients that and metabolites that your gut bacteria provides you from your diet. And so low butyrate levels have been reported previously in depressed individuals, but this link to anxiety that we observed was novel. And so how you might envision this being helpful would be if, in fact, we leverage the ability to look at the individual's microbial signature and potentially combine that with a brain image or some other phenotype. In the previous paper I told you about, it was using a clinical phenotype. We could then create some sort of an algorithm that would help us predict whether or not somebody should get one particular treatment or another. So this is sort of the main key finding that I want to send home to people at some level, is this idea of precision medicine. What it means is that we need to take into account the biology of the individual in order to understand their profile of clinical phenotype, which might be the same measures as somebody else, but could have its own unique biology. And so you've seen this slide before, but it's just to remind me that we're shifting now, talk a little bit about the types of microbiome-related approaches we might be able to use in psychiatry. So the first thing I want to just highlight, because it often is a question that comes up, is one of the things that we think in a normal, healthy profile of aging or of development is that the window early in life, that, birth till adolescence, is a bit more of a dynamic changing time. But at some point you arrive at the point where you're in your adult microbiome state, and that's pretty stable on a large scale. But in aging, then again, there's sort of a reduction in diversity and changes going on. So your microbiome is thought to be fairly stable, and that was sort of a consensus across the field. But this study that was published about a year ago looked really very closely. It was more driven to help us as scientists figure out how to design our studies. But it's helpful because if you look on the left-hand side, it shows that there's species of bacteria in your gut, some of which that upper panel shows are very stable. So if we identify these stable bacteria that represent a phenotype, then we can look at those and probably predict fairly well if they're linked to a clinical outcome. And then there's some taxa that are stable, but in different individuals, they're at different levels. And again, this is very useful if those differences land in sorting people into 2 subtypes. And then finally, there are these variable species, which they might change over the course of a day or respond to a specific diet or respond to a change in sleep patterns or all these different things that happen in the life. And so if you want to use these as your biomarkers, then you really have to have a lot more data. But the other thing about that is you have to look at it over time, which is what we're planning on doing in that cohort that I described to you earlier. Another aspect of your microbiome, which I think is really very interesting and potentially has a lot of clinical potential, is what a concept called volatility. And this came out was suggested by Thomas Bastiansen in his Ph.D. thesis, which he did with John Cryan. And the suggestion is, in this graph, you see these two different dots that are connected are two different time points where they sampled between, I think the upper panel is a group of individuals, and the second panel was a redo of the same thing. And the difference between those two time points, if they measured the microbiome and looked, was a difference in composition and diversity. So some people's microbiome was more stable and some was more diverse. But what you can see is the diversity was greater in individuals who were exposed to a stressor. So the idea that your microbiome is responding to your stress is an important feature that we might want to measure. But even within that stress group, there's some people whose microbiome was still very stable. So again, we still have to be able to see, who might benefit from a microbiome targeted therapy. Okay, so we'll talk about a few of those. So we'll start with probiotics. So probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. And the creation of probiotics, or what led to probiotics, actually came more than 100 years ago. And Eli Metchnikoff, who actually got the Nobel laureate in 1908 for the discovery of macrophage and phagocytosis, actually wrote the book called The Prolongation of Life, where he proposed that people who lived a long time did so because of the sour milk that they ate and their gut health. And so he inspired Minoru Shirota to investigate this connection between bacteria and good health. And it was actually Minoru Shirota who was the inventor of Yakult, which is a yogurt-like probiotic drink that contained Lactobacillus cereus, and it is still on the market today. And so the concept that probiotics are benefit to mental health is linked to the term psychobiotics. And this was coined by John Cryan and Ted Dinan a decade ago. And this is the concept that prebiotics, probiotics, and other substances that may exert their psychological effects via the microbiome. And so I'm not going to go through a lot of different studies that go into the details of probiotics and mental health, but I will provide these resources for individuals who are interested in looking a little bit deeper into probiotics. And the International Society for Prebiotics and Probiotics is a scientific society focused on these two areas, and their website provides a wealth of information for consumers, for scientists, for clinicians, and the consumer guides that I've put links here actually are very useful. There's the first one on the left is a Canadian one, the one on the right is a US one. But what they do is they summarize the evidence. So for every condition, they'll list the probiotics that are on the market, and then they'll provide a number, which is the level of evidence that that probiotic works for the condition listed. So if you're interested in finding out specifically about specific probiotics, these are good guides to go to. And then the other thing I will mention, I wrote a commentary on an article that reported benefits of prebiotics, which are inulins and fibers and things that feed the good bacteria. About 6 years ago, and at the time, the data was heavily reliant on animal models, mouse models, and stress models in mice. But the data at that point was quite optimistic. And now I would tell you that there is probably a good body of work done in people, both healthy and people in different clinical conditions, that demonstrate benefits to probiotics. But of course, the difficulty becomes some of the confounds of those experiments, where people, they're difficult trials to assess. But also, they have yet to take a precision medicine approach in order to determine in the front end who might be benefit from these interventions. Okay, so the next topic that I'll introduce, which people might have heard of, is fecal transplant. And so this is, as it is suggested, as shown in the graphic on the bottom right, that it takes healthy microbiota and processes in a way. And in this case, this is a New England Journal of Medicine paper, again, a decade ago, that demonstrated that if you gave a fecal transplant to somebody who had recurrent C. difficile, that it could in fact restore the microbiome and cure the infection. And so this was really the biggest, the first or the most recent advance in this space. And in fact, it was just earlier this year that the FDA actually approved orally administered fecal microbiome product for the prevention of recurrent C. difficile. And so these, what are now commonly referred to as capsules, are actually an oral pill that you can take that delivers the bacterial complex to the intestinal tract. And the idea is that by providing the recipient with a healthy microbiome, that it can not just restore the microbiome, but restore some of the benefits of a good microbiome from the bottom up, the barrier integrity, the immune system, and potentially other systems. Another popular topic is diet. And so there's been some really innovative work to try to really understand what is a normal microbiome. And this is the question that initially a lot of the work was looking at differences between healthy and some biomedical condition. But in the long run, we really do need to understand how the normal healthy microbiome works. And so this study I highlight here actually looked at 34 people, and they took stool samples and very high-level dietary records for 17 days. And they noticed that the minutia was not really the biggest factor. And so the variation was related to food choices, but not to the conventional nutrients that you might be tracking. And that in order to see this, it happened over a little longer epoch than a daily epoch. So it was more like a couple of days that things would have an impact. And most importantly, similar foods had different effects on different people's microbiomes. So if we all ate the same diet, we still would have different microbes. And if you remember from that very first earlier slide I showed about genetics and other things happening, I think I forgot to highlight, but in your genetics actually contributes to a lot about what your composition of your bacteria in your gut is and how it functions. And so, of course, it makes sense that different habitual diets might lead to a different profile of microbiomes between large populations, but each individual's microbiome can respond to very specifically to its own diet. And so if we think about psychiatry, this is a recent publication or that came out of Australia. And what they did here is they actually did a randomized control trial to look to see whether or not they could use diet to treat individuals with major depression. And again, with a very careful monitoring of diet intake, and provided people with all the foods that they needed, which is, not necessarily always applicable in certain populations. And Dr. Felice Jacka is a leader in the area of nutritional psychiatry, which has really come on board in the last 5 years. And of course, in this paper, they're pasted the conclusion here that dietary improvement may actually be a effective and accessible treatment in the management of depression. And in particular, they recommended that there could be some common comorbidities linked to potentially anxiety or metabolic health that could benefit from improved diet. And this book that I've featured here on the right-hand side is a recent book that Felice put out that is for the public, that looks at how diet can influence mental health. So again, just to remind of the point here is, if we're going to use diet, or we're going to use probiotics, or we're going to use fecal transplantation, we really do have to map out and use biomarkers that recognize the individual biology of the people in order to, have the best treatment outcomes. Okay, so I think I was faster than I expected, but most of the work that has gone on in the last 20 years is my group in McMaster, which is pictured there, and some of my colleagues who contributed to the paper I showed you from here at UT Southwestern at the CDRC. So I think that we are good for questions.
Thank you, Dr. Foster, for your presentation. We will now begin the Q&A portion of this program, starting with a few questions submitted in advance by registrants before going to audience questions in the chat. We received numerous questions in various forms about what to eat, when to eat, best forms, variances for gender and age, etc., to have a healthy gut. And here is the essence of some of those questions. The first one is, what are the best foods to eat for a healthy gut? And should everyone take a probiotic or a prebiotic?
Okay, so I think I tried to cover a little bit of that in the talk. And so, or at least it's important to understand the mechanisms of how those things are working. So prebiotics feed existing bacteria, and probiotics don't always colonize either. They tend to be a little bit more like a vitamin that they pass through and have a beneficial on the way through, although several companies are trying to develop probiotics that colonize. So most people who take a probiotic take it every day. And if you look off the shelf, or if even you look on the ones in those charts that I provided, there's often a prebiotic in there. So the prebiotics are sort of food for the probiotics. So there is evidence, and some of the best prebiotics in the world are the oligosaccharides that we get in our breast milk, right? And those feed the good bacteria right from the get-go. So there are some synthetic oligosaccharides that people have, or oligosaccharides people have developed and demonstrated there's an impact on mood. So yeah, I think that those are beneficial. Should everybody take a probiotic? It's similar to the should everybody take a vitamin question. So I'm not sure that I'm qualified to answer that question, but there are very few side effects to probiotics. Sometimes if people have GI issues and they start taking probiotics, they actually have a little bit more disturbances when they initially take them, but they settle down and often experience benefits in the longer term, because if you actually have a gut imbalance that you're aware of, or gut symptoms, then that might be a good indication that a little bit of, positive input could do. And then the what to eat. I I can tell you what not to eat, which is probably just as important as what to eat, but the best thing to avoid is refined sugar. So refined sugar does more problems for your microbiome than anything else. High fat, there's a lot of evidence in that, but high fat, as we know, contributes to all sorts of other metabolic issues in the body that might be facilitated or regulated in between the host and the microbes. But sugar would be the thing to avoid from especially refined sugar and processed foods, which is normal. Generally, people think healthy heart is healthy gut. But if you wanted to give your microbes a boost, you know, the berries and all those ones that are full of all those great you know, antioxidants tend to do your microbes good. And Felice's book actually has recipes. So fermented foods, if you're interested in fermented foods as well, fermented foods don't necessarily have the bacteria in them, but they have the products of the bacteria and they can be beneficial. So there's been people who've tested whether some people respond better to fermented foods or fermented foods with probiotics. And it seems to be that it depends on the health status of the individual, whether they can benefit from the probiotics, because if you don't have the prebiotics, if you don't have the bacteria to ferment the foods, then it's hard, you know, you might need both. Sorry, I might be a bit confusing there.
Thank you, Dr. Foster. You actually answered my second question when you talked about the sugar and things that we shouldn't eat. So thank you for that. The next question is regards to microbiota changes as women age, and does this possibly contribute to the mental issues as far as brain fog, mood swings, etc., that many women have when they're experiencing menopause?
So there is, I mean, there's a couple of different windows where there's, you know, a lot of change going on in your body from your physiological system. And one of those is during menopause, and it does increase risk of mental health issues during that period. So certainly what we know about how stress influences your microbiome and how it can impact changes that are linked to hormones and other things, that would be a factor. Unfortunately, it's a very understudied area. Aging in general is studied, and I think during menopause, generally, there's not an observation of reduced diversity the same there is about, you know, as you get into your 60s and your 70s. So I think that it's the beginning of the sort of decline when it comes to diversity. So there's, I think, a lot of different systems changing in that period of time. And I would see probably your microbiome as one of the tools in your bag to sort of help balance that out. But I don't think it's a driving force of a lot of those changes.
Thank you. And since you're talking about the age groups of 60s and 70s, I'll go to a question in the chat from Cindy. And her question is curious if there's hope for someone in their early 70s that have struggled with mental health all of their life, if they are to make drastic changes in their microbes now.
So I study mental health because I think the coping neuron is always, you're able to save it. So, you know, unlike the neurological disorders where a lot of those present after there's already loss of the cells, I think in mental health, and I'm not a physician, so I'm, you know, I'm speaking more from a biological context, is that what we can always do is slow things down or bring them, you know, back to we can't go back to being 40, but we can sort of elevate where we are through healthy choices. So even people who introduce exercise of some level or a good diet, I know as people get older, according to the literature, there's great work showing that people who live stay in their homes or live in good social and family environments tend to have better microbiome than than people in institutions. And so we think that sociability and social behavior is critical to maintaining good microbiome. So that's a little bit of a surprise that, you know, your social setting can influence your microbiome. And it might be because it reduces the stress hormones and things related to isolation that damage your microbiome. So I think both we can dampen the damage, but also boost the good stuff. So some of these probiotics and things would be good for people who are older.
Thank you. That was actually the answer to one of the questions in the chat. The second part to the question was, is there any associations between microbial floral health and autoimmune disease?
So there is a bit of evidence of changes in individuals with multiple sclerosis. And there's also, I mean, there's very few inflammatory conditions where you don't have the microbiome influenced. So microbiome are important not just for development of your immune system, but they're important for immune system balance. So I don't think I see them as a cause. I mean, I don't know the literature suggesting a causative role for them in autoimmunity, but one of the things that they're important for is they engage the T regulatory cells, which are part of your adaptive immune system that help return you to homeostasis. So when that balance is out of balance, I think it's one of the things that happens in some of those conditions. And I think the other thing that happens is when your immune system's on overdrive, which happens when you have an autoimmune disorder, then it can be producing or shifting your metabolism to produce more toxic metabolites. And so this is one place where people have identified, for example, in certain conditions, you might end up with more sort of inflammatory bacteria instead of some of those, and similar, that could look similar to the ones that I showed you in that depression cohort, that you lose some of these beneficial bacteria. So there's different ways that these can present in people. In some case, you lose a good bacteria. In other case, you get a little bit more of one that might produce a toxin, which it might not have in a healthy body, but it does in an immune compromised state. So context does make a difference to how these things operate.
Thank you. The next question comes from Wayne. Can you comment on the short-term and possible long-term effects of antibiotic use on the microbiome?
Yeah, so antibiotics wipe out the microbiome. Oh, the antibiotic. So we know a lot about antibiotics. One thing about antibiotics, the longitudinal cohort studies that have been done, where we look at antibiotic use over the lifespan, introduce a higher risk of depression. So if we just look at antibiotic use in the large cohort studies that have been done in places like Finland and other places, the number of antibiotics you get as an adult increases the risk of being diagnosed with anxiety or depression. Furthermore, if we look at the window, and there was a study recently done in the US where they really mapped antibiotic use in the first 5 or 6 years of life, and the use of antibiotics and increased, again, rounds of antibiotics in that window increased the incidence of neurodevelopment disorders in those kids prior to, becoming young adults. So there's a negative impact of antibiotics on risk factors for psychiatric illness. And that said, we know that it's always a combination of a gene and environment, right? So the triggers are different. What triggers it in different individuals is different. And then short-term antibiotics, of course, are good because we get rid of the bacteria. And so certainly there's times when that is a lifesaver. It's often recommended to put some probiotics on board, or to put them on once you've stopped the antibiotic, for sure, to help replenish the good bacteria, because the antibiotics are indiscriminate generally and kill both the good bacteria and the bad bacteria.
Thank you. Speaking of the good bacteria and bad bacteria, I want to go back for just a moment. We had someone in the chat ask if you could give us some different names of refined sugar to look for.
So white sugar in your store, grocery store, any, refined sugar is, I mean, non-refined sugar would be things like cane sugar, but any, fructose, all the stuff that's in all the candy. My brain is not pulling it out. So you know what it is, that sugar that's in it, it's high fructose sugar, corn syrup, all of those things that so in fact, it took a while before people started studying sugar effects on the microbiome, and it was shocking how it can almost in a mouse, it can get them, causes colitis in a mouse in not that very long of a time if you increase the sugar in their diet to a high level. So and I expect that it's one of those things that if you had one of those variable microbiomes would be sort of something that might be responsive to, right? So high fructose corn syrup, refined white sugar, and just a high level of sugar as opposed to other macronutrients seems to have a deficit.
Is eating yogurt as good as eating probiotics?
I think it depends on what's in the yogurt. I think you'd have to eat an awful lot of yogurt to get the same amount of probiotics that are likely in any probiotic you get over the counter. So I think that, and in fact, there's different, I mean, the important thing when it comes to probiotics, and that's why those charts that I sent are important, a particular probiotic from the International Society of Prebiotic and Probiotic, people are channeling themselves through my head to say this, but it matters that you've actually tested that specific strain in that specific condition. So it's not just because bifidobacteria happens to be a great bacteria in one version of a probiotic, it actually matters that you've grown the actual same strain that was tested. So this is where if you look at all sorts of probiotics on the shelf, there's often lactobacillus and bifidobacteria in those, but they're not all the same. And even within the bifidobacteria strain, there are some that are beneficial and some that are not. So it's important to, check the product out that you are looking at. There are some general benefits, like there are some common bacteria that are across different probiotics. And so, those ones probably are less linked to a particular source.
Thank you. Has there been a connection between the microbiome and ADHD?
I believe that there have been some case control studies showing, similar to other psychiatric illnesses, that typically developing individuals have a different microbiome than individuals with ADHD, but there's a host of other metabolic and other differences as well. And so it's hard I don't think there's been very good sort of over the lifespan studies in those cohorts, but there is yet to be any case control comparison where there's not identified differences. And part of the reason we moved to the community approach that I showed you is that these taxa or these bacteria don't live by themselves. So while some probiotics or some single bacteria strains seem to be able to have a health effect, just like sort of vitamin D might have a health effect or vitamin C might have a health effect, really in your body, they're communities and they're working together, right? And they're all the way along your gastrointestinal tract, not just where, all the sampling we do is in the stool sample. So it's sort of what's being thrown away and it's representative of what's going on in the body, but we do have to remember there's an ecology in there.
Thank you, Dr. Foster. I have another question from the chat, and this one is from Lana. What kinds of tests can patients ask their doctors to use in order to detect what good bacteria you need in a probiotic?
So I don't know that we're at that level of science, and not all physicians. I think it's good to talk to your physician about whether or not they're aware of probiotics and the benefits. Again, I didn't go to medical school, so, and I'm, now I'm in Dallas, Texas, where I don't really actually understand the whole healthcare system here myself, but yeah, I think that there are, inflammatory mediators are one of those things that go up. So people, and if you do a blood test, there's things like C-reactive protein, which are elevated in general inflammatory tone. So those are indications of higher inflammatory tone, which evidence would suggest could benefit from, a probiotic. But again, the evidence, there's not a lot of guidelines right now for specific conditions outside of what you can find in those. The best evidence is in those probiotic charts that are linked to the ISAPP website.
Our next question comes from Jenny. Does microbiome affect growth hormones?
Not that I'm aware of. And I'm not sure what growth hormones means. I mean, there's growth hormones such as BDNF in your brain, and certainly it's influenced by the microbiome, and likely other trophic factors, but I'm not sure about growth hormones if we're talking about whether we grow tall or short. I think a lot of that's genetically driven by other systems.
Okay. Is there any truth between the best probiotics if they're refrigerated versus non-refrigerated probiotics?
A psychiatrist friend who I've done a lot of family and friends things for her, and she likes to say brown bottle in a fridge just suggests that they're closer to be more likely to be live bacteria, and that is not a bad rule of thumb. But there's also several products that are made, and they're made in a sachet where it's been freeze dried, and you add it to water yourself, and those are stored at room temperature. So I think that both apply. I think that there is quite a few good products on the market, and I think if you I like the fact that all of them are listed on those websites that give you the level of evidence as well, right? So that's looking at the specific evidence is important.
Okay, one more question, Dr. Foster. And this one is, would a pregnant mother, who received antibiotics during pregnancy cause severe allergies to the baby and or possibly autism?
Wow. Okay. Let's start with antibiotics and allergies. So there is definitely evidence that you can, if a mother has, so there's allergies there's two types, right? Those that we inherit, those that we, acquire over our lifespan. And there is some evidence, it started mostly in animal models, but has been demonstrated also in people, that if you prophylactically put probiotics on board during the window where that immune system is often developing, you can reduce some of the impacts of allergy and asthma and things. So if you are doing a model of mouse where you're creating asthma and you put the probiotics on at the time that you're creating the asthma, the mouse doesn't get the symptoms later on in life. So similarly with stress in animal systems, we see that if you have a probiotic while you're going through the stress, the long-term impact of stress is not in the mouse at a later, or rat at a later time. So the idea in people is if you put the probiotic on during the stressor, you might still feel all the symptoms of the stress, but the presence of damping down the long-term changes in your inflammation might prevent some of them. And again, I put this in the caveat that these are not blanket terms that work in every single body, but their evidence is that in those situations, there are people who benefit from it.
Thank you for that, Dr. Foster. I have one last question because there's several people in the audience have asked. Could you let us know the name of the book that you had listed on the last slide?
Brain Changer. So that's by Felice Jacka, and the one earlier was Psychobiotics, which was by Ted Dunn and John Cryan in collaboration with Scott Anderson, who writes for Psychology Today. And there's another one if people are interested for their kids, which was written by Brent Finley from Vancouver, which is called Let Them Eat Dirt. And there's a few others. There's a few others out there that are targeting the public and trying to, advocate for less super hygiene and more bacteria. Bacteria is good, right? So.
Okay, I'm actually I'm putting in the chat. Everyone, if you have questions about those books, because I didn't get those written down for you, we will email you. If you'll email us at publicaffairs@utsouthwestern.edu, we will get that full list from Dr. Foster and email you so that you can have those as well. And we just want to say thank you so much. That was very interesting. I think we all want to know how our diet impacts our health, but especially our mental health. And unfortunately, we got the same answer as every other doctor gives us. Cut down the sugar, cut down the fat, exercise. So I guess we all know part of the big answer, do all those things. But we wanted to thank everyone else for joining us tonight. It was a great presentation. We want to thank our partners at UT Southwestern Harmony at the CDRC, Sierra and India on our social team, and Charlie for handling our Q&A tonight. This program concludes the spring season for UT Southwestern Science Cafe. We are currently finalizing our program details for fall 23, and we invite you back for our next Science Cafe in late August. Stay tuned for more details via email. For now, we wish you all good health and wellness and a great summer season. We are adjourned and good night. Thank you.